Welcome to MrCScience.com's video on bacteria. We're moving into microorganisms. Bacteria are extremely small, little single-celled organisms that are prokaryotes. You read earlier last module that there are two different types of cells, basically prokaryotic and eukaryotic. Prokaryotes do not have membrane-bound organelles. So they don't have little organs inside the cells that are bound in membranes. Eukaryotes do. This will become more clear as we move into our eukaryotes and our module 6, which will be our cellular biology. But that being said, bacteria are small, little single-celled organisms, incredibly small. One bacterium on average probably averages about 2 micrometers or micrometers. What a micrometer or a micrometer is 1 millionth of a meter. So that is 0.0000002 meters. Can't really relate to that, so let's make that a little more relevant. So if we go under our metric system, we find that this is decimeters, this is centimeters, this is millimeters. So that means that a 2 micrometer bacteria is 0.002 millimeters, which is still hard to relate to. So let's talk about under a microscope then. So last time, talking about microscopes, looking at them, we found that our microscope's high power can magnify things 400 times, can make it 400 times bigger. So if we multiply this 2 millimeters by 400 times bigger, we're going to get 0.8 millimeters, just under 1 millimeter. When we look at a bacteria under a microscope, it's going to look like a little dot or a little dash, or as we'll find out, little squiggle. So very hard to see under the microscope. Any light microscopes have a hard time getting there. You might be able to get some very high-tech microscopes that can look at them, and of course electron microscopes may think they're rather large. So let's take a look at bacteria. Okay, this is just kind of a general view of bacteria. Trying to draw a little bit of every possible piece you'll find on a bacteria, every organelle or characteristic. It's taken from, there's a better drawing in your book, figure 2.1 on page 38. If you want to look at that as well, or you can look at my scribbles. Okay, so this is a general idea of bacteria. Bacteria is about 2 micrometers long, so extremely small. That being said, your book kind of talks about this really well. Bacteria far outnumber every other organism on the planet. So, in fact, they say if you took just the standard mass of the bacteria altogether, they are much more massive than all the other organisms put together. So, bacteria are everywhere. They will learn a little bit about what they eat and how they survive, and you'll find that bacteria live pretty much everywhere. Vacuum of space, extreme temperatures can destroy them or actually cause them to go kind of dormant, but they're everywhere, and they reproduce quickly. We'll talk about that in a coming up portion. So let's talk about some of the parts of general bacteria. Okay, my drawing doesn't show it great, but there are generally 3 layers to the outside of a bacteria. The outermost layer is called the capsule. This is a sticky, sugary substance designed to allow the bacteria to stick to things, stick to where their food source is, where they want to be. So capsule is generally there for stickiness. Your book gives an example of bacteria on your teeth, on your enamel, clinging there to help you with some of the food that you take into your mouth. So the next layer is the cell wall. We'll learn that cell walls also exist in plants. Cell wall is a rigid material, gives the bacteria its shape. It is porous and allows water to flow through, other things to flow through it, but it does have some job in regulating water going into and out of the bacteria. So the cell wall gives it its shape. Bacteria generally come in three different shapes: a round or ball shape called coccus, a rod or pill shape called bacillus, and a helical or spiral shape called spirillum. You'll need to know these three shapes to help identify bacteria as we're moving forward. The innermost layer is called the plasma membrane. Plasma membrane will definitely cover much more in depth in Module 6. It's a very interesting design. That for sure controls what goes in and out of this cell. So the plasma membrane, also known as a bilipid membrane, keeps everything in that's supposed to stay in, lets things go out that it doesn't want, and lets things come in that it wants. So that's the plasma membrane. Inside the plasma membrane is the cytoplasm. Cytoplasm is kind of a jelly-like fluid that holds everything together, allows things to travel through it, allows some biochemistry to occur in it. Um, it's got some ions in it, it holds the nutrients and allows things to flow through it. It's kind of the stuff that fills it out, gives it a place to be. Inside the plasma membrane, and very difficult to see, although we'll talk about being able to see it anyway, things are is the DNA. DNA stands for deoxyribonucleic acid, which you've probably already learned. Um, DNA is the information holding device. It carries all the information for how to build the cell, how to maintain the cell, how to repair the cell, and all the processes that go on with it. Funny thing about DNA even carries information on how to decode itself. So interesting if you're interested in information theory, that kind of thing. I'll look into that. But DNA is inside the bacterium because the bacterium is alive, and one of the requirements for living is DNA. All these little dots I drew in here are ribosomes. A ribosome is a protein that builds other proteins. It's like a little chemical factory, and we'll learn, especially in Modules 6 and 7, that the cell relies on proteins to control what it does to make things function. So it does not have a membrane, so it is just a really big molecule, and very, very difficult to see except under extreme conditions or electron microscopes. Little hairs drawn here are fimbria. Fimbria are not for locomotion, they're not for movement, they're for clinging things. The capsule's sticky, it clings, but the fimbria also cling. And we'll talk about a process shortly here called conjugation, where two bacteria exchange genetic material. If the fimbria are used for that, they have a different name. They're called sex pili. They say it's under reproduction. Not really reproduction, it's more exchange of information, but we'll talk about that more very shortly. But so fimbria or sex pili are not used for locomotion, they're for clinging to substances or other bacteria. The thing that is used for locomotion is flagellum. If the bacterium can locomote, can move, it has to have a flagellum. That's the only way it can do it. Not all bacteria have flagellum. Some will have more than one. Flagellums are kind of interesting. It looks like a tail here, but really what it is a little rotary engine here with a hook or with a little elbow in it, and spins that elbow around, and that tail flops around, and it generates drive. It allows it to move. Not very exacting. It's mainly gives it a general direction, but the bacterium just wander. Flagellum are very interesting. If you want to look into the world's smallest complex rotary engine, look into flagellum. I wandered over to Answers in Genesis site and borrowed one of their images. I'll put it back when I'm done. Of the flagellum that sits inside the plasma membrane. It's got a little rotary engine here. Here's that little elbow or universal joint they call. It's got some bushings, it's got everything you look for in a rotary engine. Scientists still don't completely understand how this operates. So there's something to look into. And one thing they definitely don't understand is how it could have evolved. Too many working pieces and too many things required for it to work at once, that if it evolves slowly, it would work at all. So, if you're interested in that kind of things, I recommend a book by Michael Behe called Darwin's Black Box. It's a bit technical, but it's a very interesting read. And one of the things it talks about is the flagellum. So, we got our bacteria. Know your parts of your bacteria, know your shapes of your bacteria. And if you have any questions about any of this, as usual, contact me at Mr. C at MrCScience.com. Thanks.